So I want to start with this graph showing that we're living in unprecedented times when anthropogenic climate change is happening now, it's happening rapidly. And because of that, we need to understand how it will impact the ecosystems on which we depend. So most of us have probably seen a graph like this before. I should have asked earlier, can everyone see my mouse if I use my mouse to gesture? I'm not sure.
Yeah, we can see it.
Okay, great, thank you. So a lot of us have seen graphs like this where we can see the rapid rise in temperatures over the last few decades. But the reality of climate change is not a single squiggly line. The pace and magnitude of climate change, so how rapidly and how much it's happening, varies across space and time. So different locations are experiencing different rates of warming and climate change, and that has implications for understanding how species and different communities will be impacted by climate change. So usually the Hawaiian Islands are, off to the side here, sometimes not even visible. I really like this map that I found when I was doing my PhD research, and I don't even remember what textbook I found it from. But in this map here, Hawaii is right smack in the middle. And you can see that it's a really special place because it's very isolated, surrounded by thousands of miles of the Pacific Ocean. So that means that it's very sensitive to climate change, and in particular, climate change associated with variation in Pacific sea surface temperatures. The other reason why it's such a special place to work is because of the large number of endemic species, so species that occur on Hawaii and nowhere else on Earth, and the number of species that are also endangered. So all important reasons for doing research here. So today I'm going to talk about climate and vegetation change over 3 time scales. First, over centuries in the Waikapū ahupuaʻa on Maui that occurred after initial Polynesian and then later European arrival using fossil pollen methods. Then I'm going to talk about vegetation change over decadal timescales across the Hawaiian Islands during an El Nino event and then over the past 4 decades using satellite observations. And then I'll end by talking about seasonal phenology from the Laupāhoehoe wet forest plot and the Pālamanui dry forest plot on the island of Hawaii using phenology data from litter baskets. So the first part of my talk, I want to start by giving this background. So you can see in the bottom here, I have a few references. In the 90s and early 2000s, there was sort of a consensus that formed around how we understand Polynesian impact on Hawaiian vegetation. And lowland vegetation today was thought to be nearly completely altered from pre-human vegetation, primarily because of the use of fire to clear land for agriculture by early Polynesians, as well as the introduction of non-native species. Fossil pollen studies indicate that vegetation change occurred rapidly after Polynesian arrival. And in some cases, vegetation change seemed to occur before even the evidence of fire. So possibly suggesting a role for the Polynesian rat that came predating on native seeds. So we can look at, sorry, I just saw this chat. I'm not sure if we should look at it really quickly. Okay, sorry. So we can look at vegetation and climate change in lake sediment cores that preserve a timeline of change. And this is the picture on the bottom right here. This was during my PhD work at UCLA, where we're manually hammering one of these sediment cores into a pond on Maui. On the left here is an example. This isn't our core, but this is an example of what one of these cores look like when you pull them out. And the basic idea is the top is more recent in time and the bottom is further back in time. Preserved in these sediment cores is fossil pollen from different species. And if you look at the pollen under a microscope, different plants, if not species, and at least genera, families, groups of plants, they look different. Their pollen types look different. And so you can look at the abundance of pollen from different species. And my examples here on the top right, this is what pollen from ohia looks like. And on the left here, this is pollen from an herbaceous species that indicates more open non-forest conditions. So you can use fossil pollen to track vegetation change, and you can also look at other indications in the sediment record, such as charcoal, which can tell you about the frequency and the intensity of fires going back in time. So we took a sediment core from Kealia Pond. Here's a map showing Kealia Pond here, and it drains a pretty large drainage basin. So this is where we took a sediment core. This was back in 2009, I believe. And that sediment core is going to contain a record of vegetation and environmental change from the vegetation locally surrounding the area, but also through this entire drainage basin. So it's also tracking vegetation change from higher elevation areas as well. So here is a figure showing that vegetation change, and I'll walk you through. There's a lot going on here. So on the y-axis, this is time, going back in time. Our sediment core goes back to 650 BC, and at the top is more recent. And on the left here, this is the change in pollen from forest, tree, and shrub species. This is how the pollen has changed through time from an herbaceous group of species, indicating more open conditions. I'm not going to talk about pollen accumulation rate. Here, this is a change in charcoal, again, indicating changes in the frequency and intensity of fires. This right here is a precipitation reconstruction based on the pollen. And on the right here is an independent measure of Polynesian population sizes. So the first thing that I want to point out is if you look at the charcoal record, there are fires that seem to be occurring further back in time, way before Polynesians would have first arrived to this area. But there is a really strong peak here, right around 840 AD, and this aligns pretty well with this independent record of Polynesian population growth. And the arrival of the Polynesians to this region on Maui appears to coincide with this shift from dry to wet conditions. So if we look at this precipitation reconstruction here, you can see that it's generally wetter here and generally drier here. Another indication that it was drier during this period and wetter up here is the presence of gypsum. And gypsum is a mineral that forms during dry conditions that we found several times in the sediment core prior to this period. And this period is called the medieval climate anomaly. So the medieval climate anomaly was a global event. In the Pacific, it's associated with what's thought of as fewer El Nino type events, and that has to do with variation in Pacific sea surface temperatures. So the Polynesians seem to arrive to this region on Maui around 840 AD, and it coincided with the shift from dry to wet conditions. Here's another graph of the pollen, and there are a lot. So again, this is y-axis is going back in time. The top is more recent. All of these right here, which might be hard for you to read, these are just different plant species or genera at least. And so on the left, these are different ferns, these are trees and shrubs, and on the right are herbs and grasses that indicate more open conditions. So you don't need to be able to read all of the text on here, but I will point out for you the main takeaway from this reconstruction of vegetation change. So how severe and how rapid did the Polynesians alter native forest diversity? This is the Polynesian period right here. And native trees and shrubs remained diverse for about 600 years of Polynesian presence. The greatest reduction in pollen diversity occurred during the European period. So post 1778 is when we see the biggest reduction in the different types of pollen in our sediment record. Once Europeans came, we see that montane forest taxa began to decline. Native lowland taxa disappeared almost completely from our record. And we began to see the presence of non-native invasives such as Kiawe in our pollen record. So this is what I put up earlier. This was the general consensus that people had formed in the 90s, early 2000s, and I'm going to have that dissolve away. Based on my research, Polynesian impact on Hawaiian vegetation was not as severe as previously thought. Early Polynesian vegetation change was probably confined to the lowlands. At this site, in my study, there was no evidence of rapid and extensive forest clearing immediately after Polynesian arrival. And in fact, the greatest decline in native forest diversity occurred during the European period. And the other thing that I showed is that vegetation change is sensitive to variation in Pacific sea surface temperatures, such as El Nino type events. So the second part of my talk, I'm going to talk about decadal changes in vegetation across the Hawaiian Islands using satellite observations. Satellites give us a really unique perspective on ecological processes at the global scale. And so here what we're looking at on top was a study showing this sort of global greening. And there have been more recent studies also suggesting that there have been long-term greening trends across the Earth. You can see that pretty well right here in the Amazon basin. So it seems to be that tropical forests in particular are showing this greening up from satellite observations. On the bottom here, they've hypothesized the three limiting factors to vegetation in different regions. So for example, in higher elevations, higher latitudes, but also higher elevations. In higher latitudes, it makes sense that temperature would be more limiting to plant growth and plant productivity. In drier regions, such as the Southwest in the US, for example, water may be more limiting. And then in tropical regions, because they're generally warm year-round, which is favorable for plant growth, and they generally receive plenty of sunlight, so, long days. And they generally receive plenty of precipitation in tropical regions. They are thought to be most limited by light availability. So even though they experience these long days, there may be changes associated with cloud cover that might impact the growth and productivity of these forests. So one example of this came from the Amazon. The Amazon experienced a really severe drought in 2005. On the left, what you're seeing in red are negative precipitation anomalies. And that just means in red, these are lower than normal rainfall values. On the right, this is a satellite index called EVI. It's a satellite, what we call greenness index, which I'll talk about in a minute, showing that during this drought, when there was less rainfall than usual, there was also an associated greening up of the forest. And so these authors hypothesized that during this drought, there were fewer clouds and more light was able to allow the plants in that tropical forest to be more productive. So greenness, earlier I just showed an index called EVI. One of the most common satellite measures of greenness is called NDVI. EVI is also really commonly used. They're similar in that they're both based on differences in reflectance of the vegetation across the electromagnetic spectrum. So NDVI is very commonly used and we call it greenness. What it is though, it's a ratio between the reflectance of visible and near infrared wavelengths from the surface of vegetation. So green, healthy vegetation in general, it's going to absorb a lot of the radiation in the visible spectrum, and it's going to reflect a lot of radiation in the near infrared. In contrast, brown vegetation, so vegetation that's dying or maybe going dormant, senescing as we would say, it's going to absorb less in the visible region, and it's going to reflect or transmit more in the, less in the near infrared compared to healthy vegetation. So it's this ratio between visible and near infrared that's telling us something about the productivity and the growth of these plants, and it's generally referred to as greenness. So not every tropical forest is the Amazon, and of course there's a diversity of different tropical forest types. So across the Hawaiian Islands, there are these large climatic gradients within a small geographic area, so we can look at how different tropical forest types respond to drought. And so this was, again, a part of my PhD research at UCLA a long time ago, where I looked at how these islands respond, how the vegetation on these islands responded to an El Nino that occurred in 2002 to 2003. El Nino conditions generally result in drought for the Hawaiian Islands, and that's for a couple of reasons. The first is that during an El Nino, which is defined by warmer than normal equatorial sea surface temperatures, during an El Nino event, the trade winds weaken, and we know that the trade winds bring a lot of rainfall to especially the windward side of the islands. So the trade winds are weakened. There are El Ninos are also associated with a strengthened Hadley cell circulation. So when sea surface temperatures are warmer than normal, that drives this atmospheric circulation more strongly. And Hawaii is sitting somewhere in the subsiding zone of the Hadley cell. So that strong atmospheric subsidence is associated with a high pressure system, and that's generally associated with warm, dry conditions. So here what I'm showing are correlations between NDVI, our greenness index from satellites, and an El Nino index. So the blue colors are showing a greening up that occurs as this El Nino event gets stronger. And the red colors are showing a browning down that occurs as this El Nino event gets stronger. And I always look back and think that if I was smarter, I would have made brown down brown and green up green. But instead, just remember that red is a browning down, blue is a greening up in response to this El Nino drought developing. So if you look at this map and it's not super interesting to you, I would agree. But what happens when I start to lag the the NDVI behind the El Nino sea surface temperatures, you start to see a more interesting pattern emerge. So you can see at 7 to 8 months, there is this divergent response, and it shows up especially well on the Big Island, where the windward side is showing this greening up in response to this El Nino, and the leeward side is showing a browning down in response to the El Nino. And I should also say that I realize not all of these regions are vegetated, and so in the paper itself, we do look specifically at vegetated regions. So this is suggesting that wet forests are greening up, while the dry forests are browning down. Fewer clouds, suggesting fewer clouds, more light, and more green on the wet forest site, and perhaps the dry forests are more water limited. And I should also add that we did do additional statistical analysis looking at rainfall patterns and cloud cover as well. So this is an updated analysis, and this is looking at long-term greening and browning trends across the last 4 decades. This is work that's led by Austin Madsen. He's currently at the University of Wyoming. This is again looking at NDVI, that greenness index, and this is just looking at long-term trends in NDVI. So you have to first orient yourself a little bit and tilt your head. This is the Big Island here. We have a north arrow down at the bottom, so north would be pointing this way. This is looking at the full time series from 1982 to 2019 in this NDVI time series. And you can see that there are regions on the windward side. Oh, I should also say it's in the legend here, but the light gray pixels are native vegetation pixels from a land cover map. So you can see that there's some green here showing long-term greening on the windward side of the Big Island, and then the red pixels are showing long-term browning. Then we can look at particular months. So this would be every January from 1982 to 2019, every February, et cetera. And when you look at particular months, you can see during certain months, May, for example, this divergent response, again, especially on the Big Island, of greening on the windward side and browning on the leeward side shows up really well. So what's driving that? Here on top, we're looking at a map of precipitation changes. And so the darker the red is a more negative trend in rainfall over these last 4 decades. So we're seeing a long-term decrease in precipitation, and that shows up especially well on the windward side here. And then on the bottom, again, is the correlation between NDVI, and precipitation. And again, we see, especially on the Big Island, this divergent effect. So red meaning less rainfall associated with higher NDVI associated with the greening. And on the leeward side here, green colors indicating less rainfall associated with browning. All islands showed a 33% or greater decline in NDVI over these last 4 decades, especially the island of Hawaii, where there was a 44% decline in NDVI. But we did see greening in some windward areas, and so that sort of corroborates that earlier study of that 2002-2003 El Nino event. Declines in NDVI can indicate canopy volume loss, it can indicate declines in productivity, and it can indicate tree mortality, for example, or vegetation mortality. And this can be possibly further impacted by the lack of regeneration due to grazing from goats, cattle, and other non-native species. In the future, what this suggests is if, and it's a big if, the drying and warming of the climate on the leeward slope of the island continues, native ecosystems may be increasingly vulnerable to fire and be replaced by invasive species such as kiawe. And I say big if, not being skeptical of climate change, but just trying to understand, again, the magnitude of not just warming temperatures, but associated precipitation changes in different regions across the island. So this last part of my talk is going to discuss seasonal tree species phenology from Laupāhoehoe and Pālamanui, using phenology data from litter baskets. Changes in phenology, so this would be, for example, patterns of flowering and leafing. These are some of the most sensitive responses to climate change. So people refer to phenology often as nature's calendar, and it's defined as the timing of biological events, such as when trees bloom, when they drop leaves, or it also refers to animals. So for example, when the humpback whales return to the Hawaiian Islands in the winter. One of the strongest, what people say, fingerprints of climate change. So one of the strongest impacts of climate change that there's consensus around is what's called spring advancement. And that's an earlier occurrence of spring events such as bud burst, flowering, and migration of different birds and insects, for example. So here, this is an example of what people typically think of when they think of phenology. As the climate begins to warm, moving into the spring, coming out of winter, we begin to see trees put on leaves, and then they might flower, and then there's peak greenness or peak growing season at some point. And so when we think about spring advancement, the earlier occurrence of these events, it can be really disruptive to a lot of ecosystems. And the analogy I use is, for example, if you think about your work week and all of a sudden Monday came earlier, so you didn't have a weekend anymore, and before you were ready, it was Monday and you had to get back to work. That can be really disruptive to you. And that's similar to what's happening with these ecosystems with climate change. Most of this evidence of spring advancement, however, is coming from Northern Hemisphere temperate and higher latitude boreal or Arctic regions. This down here is a meta-analysis I was a part of where we showed the negative numbers here are indicate in advance in each of these events. So for both flowering and leafing, using several long-term phenology data sets, we show that they were advancing between 5 to 7 days earlier per degree Celsius of warming. The thing is, in the tropics, there's no spring advance, right? So tropics don't have a typical spring like these temperate regions. On the right here, this is plotting the climate space of where a lot of these long-term phenology records are from. So this is mean annual temperature on the x-axis, mean annual precipitation on the y-axis. And in both the gray and the blue dots are where most of our long-term phenology records are located in climate space. And they're all located in these generally like temperate or higher latitude climates. This right here is one tropical site. This is Luquillo in Puerto Rico. Okay, so long-term tropical data sets are really rare. What that means is that we know very little about climate sensitivities beyond temperate mid-latitudes and beyond these higher latitude systems. This graph right here is showing the number of species that are responding in their first flower or first leaf date to temperature. And then this is plotting the number of species at these sites and the mean annual temperature at these sites, okay? So what I want to point out here is this is our tropical site that we included in this study. And you can see that in general at this tropical site, but also as sites get warmer across all of these data sets, fewer species are responding to temperature in their phenology. So this suggests that tropical species may instead be responding to changes in other climatic factors, not temperature, but maybe precipitation or changes in sunlight. So how is Hawaiian plant phenology responding to climate change? And what does greening and browning mean on the ground? So here I'm going to present some data from ground-based phenology records from two contrasting forest plots. Laupāhoehoe is our wet forest plot. It's dominated by ohia and koa. It receives plenty of precipitation year-round, more than 4,000 millimeters of precipitation, no distinct dry season. And the dry forest site is Pālamanui on the Kona side of the Big Island. It's dominated by lama and alahe'e. It receives much less precipitation, about 800 millimeters mean annual precipitation, but also a very strong summer dry season. And then on this figure here, this is just a picture showing in ohia Laupāhoehoe and a lama at Pālamanui. So we collect phenology data in these litter baskets. On the top left is an example of one of these litter baskets. There are 64 of them set out in a 4-hectare plot, and each plot in sort of a grid pattern. I shouldn't say sort of, in a grid pattern. And every month, all of the litter from these baskets are collected and dried. The leaves are weighed. So we have a measure of leaf mass every month. Only in recent years have we been sorting leaves to different species. The seeds are sorted to species and they're counted. And on the top, what you're seeing is an example of what we found, just an example of what was in one of the litter baskets in Pālamanui. So you can see the different, those are llama flowers and llama seeds, for example. So those would get counted, and then the different leaves as well would be weighed. So I do want to point out the hard work of all of our field techs. There would be no data without these ladies and other field techs that have worked with me throughout the years. They are hard at work going to both of these sites once a month, drying, weighing, and sorting through all of the data. So in the analysis that I'm about to show in the next few slides, there are 65 months of data from the wet forest site from Laupāhoehoe, but we are continuing to collect data right now. So Amberly is our tech right now working hard, and we have data right up until this month, for example. At the dry forest site, there are 36 months of data. Okay, so this graph, I'll just say, looked a little bit different in my PowerPoint, but that's fine. You can still generally see what I wanted to show here. So this is the average monthly leaf litter fall phenology from the dry forest on the left and the wet forest on the right. And one thing I do want to point out is at Pālamanui, we do have a shorter record and we do have some missing data. Okay, so we have a much better record from Laupāhoehoe. But what I want to point out is that even though we think of, especially the wet forest side, we think of these forests as evergreen, there is seasonality in their leaf phenology. So they are responding to seasonal changes each year. So here what I'm plotting is the leaf litter fall across the y-axis against these two satellite measures of greenness, NDVI here, EVI here. And these two are the dry forest site, Pālamanui. These right here is Laupāhoehoe, the wet site. What you're seeing are two distinct relationships with satellite observations. And I should say two distinct, but generally poor relationships with satellite data. So at the dry site, what's happening, well, what seems to be happening is when there are few leaves here, you can get greenness values that vary by a lot, okay? Whereas on the wet site, what you're seeing is across different levels of leaf mass, so few or many leaves, we're seeing that the satellites are telling us it's generally always green, okay? So here at the wet site, there might possibly be at higher leaf litter values when there are more leaves, there might possibly be kind of a weak correlation with a lot of variability at higher levels of leaf litter fall with NDVI. But in general, there's a lot of questions here about why the relationship is so poor and what these satellite measures of greenness mean on the ground. There's a couple of ways of thinking about what might be going on. And the first obvious question is, how well does leaf litter fall correspond to what's actually on the canopy? So there might be a time lag that we can look at with leaf litter fall and the satellite data. And I did look at some different time lags, but a lot of that wasn't super promising. There are these phenocams mounted at both sites, and these are just mounted cameras, and you're seeing two images. This is two images from Laupāhoehoe from two different dates. So on the left is a image from June, and on the right is an image I grabbed from 2 days ago. And you can see that there's variability in what is on the canopy just from these two different months. So there is a koa right here, and you can see right now there are fewer leaves, and you can see the seed pods as well. And then back in June, this is that same koa, and that canopy is much more green here. So can the phenocam help us better understand the timing of leaf litter fall in those baskets compared to what's actually on the canopy? And are there certain species that might show better relationships with the satellite data rather than aggregating all the leaf litter data? And so these are new questions that my PhD student, Craig Anderson, and postdoc Shannon Bayless are starting to investigate. So what about relationships with climate? And here I'm just going to show data from Laupāhoehoe because, as I mentioned, the data from Pālamanui is not as good with missing data. So at Laupāhoehoe, oh, shoot, sorry. Once again, you're not seeing these 95% confidence intervals that just aren't showing up on the slides for some reason. Anyway, what I'm showing here are the relationships between leaf litter falls now on the y-axis and climate is on the x-axis. So we have temperature, rainfall, and PAR is an acronym for light. So that's photosynthetically active radiation. It's just the wavelengths of light that plants use to photosynthesize. So the approach that we took here is we used a model selection technique where we compared all possible combinations of temperature, rainfall, and light. And we let something called AIC select the best fit model to the data. This approach tends to penalize for more parameters. And what that means is it wants to pick a model with fewer climatic variables in it. Okay, so it wants to be more what we say parsimonious. So we started with all three, and then it looked at every possible combination. So for example, just temperature and rainfall, or just rainfall and light, for example, and then even each one of these alone. So all possible combinations. The best fit model turned out to be the model that included all three climatic factors, even with AIC wanting to penalize for more parameters. So suggesting that all three are really important for understanding leaf litter fall. And what you're seeing at Laupāhoehoe is generally leaf litter fall is decreasing as minimum temperatures get warmer. It's increasing with more rainfall, and it's generally increasing with photosynthetically active radiation, more light levels. In some of these graphs that I'm going to show, you'll see these like sort of wiggles at the end, and that's where if the gray 95% confidence intervals were showing up in this presentation, you'd be able to interpret that better. So a lot of the like nuances in the squiggles here, we can't interpret too much into that. All right, so this is looking at the average monthly seed phenology, and these are for 4 dominant species that occur at Laupāhoehoe. So there's 21 species at Laupāhoehoe, 12 species were present in at least once in our litter basket collections, but there were 4 that were dominant and occurred frequently enough for us to perform statistical analysis. So what you can see here in Koa, Ohia, Olapa, and Pilo is that there is seasonality in seed production as well, and Pilo might be one of the most seasonal with a really strong season of seed production, sort of at the end of the summer, moving into the winter here. So once again, for seed production for all 4 of these species, the best fit model included all 3 climatic predictors. And I'm not good, let me just look at the time really quickly. So I won't go over the exact relationships with every single species, but you can see here, Koa is showing in general declining seed production with increasing temperatures, increasing seed production with rainfall, and increasing seed production with more light, different relationships with Olapa, and then again, different relationships for Pilo and Ohia. But all 4 species wanted all 3 climatic factors to best explain the variation in seed production each month. So the takeaway, without me going into every single relationship, is that this suggests that the forest will not be impacted uniformly, that all of those 4 dominant species are responding in different ways to changes in climate, to variation in climate. So suggests that the forest will not be impacted uniformly. There will be winners and losers as the climate warms. So for example, with increasing temperatures, those models I showed suggested that Koa, Ohia, and Pilo will decline in their seed production. If it gets wetter, then Olapa will decline in its seed production. And if there is more cloud cover, and again, these are big ifs, right? But if there is more cloud cover, we can try to make predictions about how these species may be impacted. So Koa and Ohia would see reduced seed production with more cloud cover. The big question is, if some of these don't seem to align with what you see on the ground. Oh, and I should also say at this site. So for example, we know that Ohio, for example, occurs in really warm lowland areas. And so at this montane site, at least, we're seeing these patterns. Okay, the last point I want to make here is that how these climatic variables interact. So for example, what happens when it's warm and wet versus warm and dry? That we don't know. And that in reality is what's happening. But we need more data to statistically tease apart those interactions. So we're continuing to monitor the phenology to try to better understand how these species respond to climatic variability. I just have 2 more slides, or plus I think 1 more slide. I just want to show some current work that's being led by my postdoc, Dr. Shannon Bayless. So given that I've just showed that these forests will not respond uniformly, current work is investigating variation that we see across the site and different vulnerabilities among species to climate variation. So in collaboration with Eben Broadbent, who's at UF, we are flying a high resolution drone over each of these sites, and it's giving us really high resolution imagery, including hyperspectral and thermal imagery. So this is just an RGB, but you can see these canopies really well, and you can start to see, so this is an ohia, and you can even see the flowering on this ohia, and you can see the koa here, for example. So we can start to look at what proportion of these different species account for what we see from the canopy. So what a satellite would see, essentially. This is a picture of Shannon right here. She's mounting a phenocam at one of our sites. And so those phenocams and those questions I asked are also a part of the current work that we're doing. And then really quickly, I'll just show a thermal image. This is from Laupāhoehoe, and this is canopy temperatures across Laupāhoehoe. And this is what one of the things she's been working on is delineating each of these crowns, identifying which species they are. And there's some really promising work looking at how canopy temperatures differ for each species and that variation across the plot. So I will end there. I think that is pretty close to 40 minutes. I just want to acknowledge a lot of co-authors, a lot of field techs, a lot of people that have worked with me. Any papers or data that you're interested in, you can find on my website, or you can email me, and I will stop there. Thank you.
Great, thank you.